Dr. Muthuvel, head of the division. Uh, my colleagues and my classmates, students, thanks to the dean and the university for giving a chance for coming. I have already come in 2014 to 19 once for the same qualifying, but that time I didn't take any guest lecture. So Dr. Muthuvel asked me last week, so I thought, okay, coming there, so I can share my experience where I did some work and did something. So this is my last 10 years of research work at IRA. So on breeding, and we did for both for varietal development for both wilt and for a high yield variety. But I'm just going to present for a yield and other purposes only. That wilt is a little bit complications are there. So we are trying. So for the past 2 days, we are discussing with the students about what they have studied. Now the students will ask what I know. Huh? Okay, so whatever it is there, so I can share my experience. That is my thing. So, but overall, my view is before starting this, students are preparing well, but don't be afraid of the board. Whatever the board is there, so you should be know that you are confident of that. So that much you should go with the confidence and be cool enough. I think one or two students are a little bit tensed. Otherwise, most of the students answered in very well. So whatever the forum you are going, this is a just qualifying exam is to have a board exam for preparation of next interview for your selection or something like that. This is just a scrutiny because writing part you will have, and for oral interview it is a part where the marks matter mainly major. So written part everybody will have it. Only all good students can score in a very minimum range, but interview matter one part of that. So this is just a beginning for you to cope up further in your career. Most of the thing it went well, I think. Okay. So just while going through this, as we know that guava, what are the approaches we have in breeding? Normally we go for introduction, selection, hybridization, then mutation breeding, then we will go for evolution. And so some of the varieties I will show, some of the species I will show what are the characters, and we can go for some of the improvement. So for suppose we are going for some of the molecular we discussed for the past 2 days. So here we can go for some of the screening work where we can identify. So suppose I have these are some of the part of the students' work also. My MSc student Shaili Kumari did some work. Then my PhD student Nayan Tibek did some work. So all the work wherever, because why I am saying is that student force is one of the major contribution for the institute development, especially for master and PhD students. So we work along with the students. So students are the main younger generation to know, and they should help us to improvement of the division. Division will have a direct to the colleges, college will have to the university, and university will have a impact to the farmers. Ultimately, we want to develop a variety related to that region. So we are having how many agroclimatic zones? Huh? 7 or 8? Huh? 7 only, yeah? I heard it is 8. Okay. Hmm. Okay. So depending upon the different then after selecting that, we will release a varieties related to a first zone or state. Then after that, it will go for all India coordinate research projects. Then variety will be evaluated in multi-location. So once multi-location it is evaluated, then it will be released for at least more than two states. If it is, then it is called a central variety release proposal. These are some of the things. So before we are going to go off breeding, so why it is important? So these are the states where we have area, we have production, some of the things. So in Bihar is one of the area, Chhattisgarh, then Haryana. So here when we talk about other states called Maharashtra, Madhya Pradesh. So these are some of the area where we have more guava growing areas. Then if you talk about other major states, that one is Uttar Pradesh. then Tamil Nadu. So here Madhya Pradesh, Uttar Pradesh, Tamil Nadu and West Bengal. So these are some of the major areas. So now Rajasthan is coming very well for Goa, but naturally, normally Rajasthan doesn't have this crop. Now it is performing very well after this Indira Gandhi canal projects were developed. So we need some irrigations. So after that, because in arid crops, what is the main advantage? If you have a crop, it is under stress. So the quality of the fruits will be more high TSS. Then there will be less pest and diseases. So that are some of the special quality of drought condition, and especially in Rajasthan. So what are the districts that are under desert? Bikaner is one desert. Barmer, Jaisalmer. These are some of the deserts. So wherever we have a stress conditions and we have a higher quality, either for market or for in either terms of TSS. And another one is that major pest and diseases, soil-borne diseases will be less because it is mostly sandy and more sandy soil. So most of the soil-borne diseases will be very less compared to other waterlogging conditions. So this is one of the advantages. So these are some of the trend. If you talk about the 1987 to 2022, so the area under Goa cultivation is increasing. But when we talk about productivity after 2015, we can see it is a constant. So we cannot increase the yield or productivity, but we can go for different density planning, high density planting, pruning or manipulations are some of the things. So now the target is only for quality and processing traits. Maximum yield potential achieved can be done only by not varietal development, by alignment, spacing. You can go for ultra density planting or planting. So per acre or per hectare area can be increased. So that is the thing. So productivity is now reached maximum, but we can try if it is some of the molecular tools are applicable and we can produce some more better, but that's very, very meager. So whatever the variety we have, we want to improve the quality, either in content of antioxidants or red pulp or the content in the red pulp. It is lycopene. Huh? So we want to improve. So when lycopene is there, in some variety we found that vitamin C's are less. So we want to have that hybridization so that the vitamin C will be increased. Normally, as per my research, we found that white varieties are having more vitamin C content when compared to red. And in that also white variety we have more variant. Allahabad Safeda is having highest, Lucknow 49 is having highest, and Panch Prabhat is a white type, but it is having the lowest type, like that. But basically, when we categorize these two, is having higher vitamin C content when compared to red. So these are some of the protections we have given. So when we talk about guava, we should, as a breeder, we should know where it has come. So it is a theoretical one. It is tropical America. So how it has been introduced to India and some of the things. So these are some of the, when we talk about breeding, we should also have very, very important for the species, or what are the species, what is the character it is resistant to and how. So then only we can go for whatever the desired character. Suppose I want to have a nematode resistance. So if any species is having, because cultivated species doesn't have that quality. So when we talk about nematode resistance, so we have Prestianum. So it will be having resistant to root knot nematodes and wilt. So when we use this select as one of this parent, then we can transfer. Okay. So like that. So first, whenever we want to go for a breeding, what are the species, what are the things are there? So what are the quality characters? So suppose when we talk about cold resistance, so North Indian conditions, if we want to grow, so we will take cold resistant as one of the species for crossing. So suppose some of the interspecific hybrids are there for wilt resistance. So what is your target? Whenever we take, so what is your objective? So that's why when we design a thesis, today one thesis was there for Jamun dwarf. So what was the problem in that? Because it is a big tree and main problem is in Jamun is harvesting. Normally they will climb or they will shake on the whole tree, putting some leaves or some cloth. They will. So after falling down, the fruit will be getting damaged. So when we go for a objective of dwarfness, in what way it is going to helpful? So like that, when we found here, again this pumilum crosses is having dwarfing effect. My question is that in case of Goa, whether we need a dwarf rootstock is required, if required, or if required means why, and if it is not required means why. I say at present situation it is not required because we have now high density planting. And another thing is that guava is a what season flowering crop? Current season flowering. So if you prune, then it will come, a new shoot will come, and flower will come. So our main purpose is to have a productivity. So if it is there, why we wait for a dwarf rootstock to develop, and after that graft compatibility will there. So another problem is that when we talk about developing a rootstock, next problem is one is graftability, another one is multiplication of the rootstock. These are the drawbacks that we have, especially in guava. So normal cultivated species are having easy to graft or easy to multiply, air layering or cuttings. In seashore area, we have that cuttings, three node cuttings are there. It is very easy to root. When we have high humidity, so it is very better. But whenever we talk about this rootstock of resistance or some of the qualities, it is very, very difficult to have a rooting. So when unless it is largely multiplied and it is graft compatible, then we cannot have a successful implementation to the farmers. So the main purpose will be defeated. So in that case, for dwarf purpose, my view is that it may be changed depending upon the purpose. So my view is that when we have that pruning technology standardized and we have high density planting, then we can manage the canopy with pruning and other irrigation management. Then it can be have a better yield rather than we were waiting for a dwarf and then to get it graft. So these are some of the things. These are some genetic resources. No? So in NPBGR we are having for all crop in India we are having NPBGR as a conservation, genetic conservation and thing. And we have these are the major centers we are having working. So universities we have Basti in UP. So this is we have so Lucknow our Central Institute for Subtropical Horticulture. So we asked one of the students where is the NAC National NACS accredited centers for conservation, NAC for conservation of the varieties or species or that. So in some of the crop we have some of the centers. Okay. So then some of the horticulture experimental station in Saharanpur, then NDUAT, now it has been changed. So this is in Sangareddy. So these are some of the universities where we have. Sorry. So these are some of the. So when we talk about that, some of the organizations. So we have earlier this much centers are. Normally we don't concentrate on private companies. Why? In fruit crops only there is only public sectors are working. One or two company rarely that VNR company has been released by one private firm and now they are doing for papaya. So other fruit crops only public sectors are doing. Why? Because they don't make much profit in fruit crops. So that is the why only the sector where we are having breeding is in fruit crop is public sector. So that's why there is no competition in private sector. One or two are there, but especially for papaya resistance type like red lady or any other type. So again that VNR are doing for papaya also. So only the fruit crops where they have seed where it can be sold at a cost. So there only. Otherwise it is 200 rupees, 150 rupees graft and they don't want to invest crores of rupees. Whereas in vegetable and floriculture, it has a much demand and they make an hybrid and every time they have to go for the material. So for us, we go for a clonal propagation. So once you take for 1,000 rupees, we will multiply and we will sell. We will not go to the company. So for that purpose, they are not having much profit. So in that only reason that fruit crops are only public sector undertaking or government institution are making some of the efforts to improve the varieties. These are some of the so IR also we have. So in that when we talk about this Anupleyadi, so this one we have released this latest variety in IR, Pratiksha and Pusa Arushi. So I am also one of the breeder in these two varieties. So here Pusa region, this is the root stock. the first rootstock variety in fruit crops in India, except grapes. So we have not have a grape rootstock in India developed by India, but we have grapes rootstock. So this is the variety where we have dwarf rootstock, Pusas region. But the main problem in this rootstock is the same what I told. It is dwarf, resistant to wilt, but for multiplication of the rootstock, we faced very difficulty. Either you go for air layering, after developing rooting also if you plant establishment is very poor, then graftability is compatibility is also a problem. That's why Pusas region is not that much successful now. So our breeding objective now we have changed because we will impart this character, resistance character to the traditional varieties where we can multiply as such. So where the so that work is being done by Dr. Vasu in IHR Bangalore, which I am not going to present here. So these are some of the photographs that we talked about, some of the genetic resources of wild. So where it can be there. So this is Pseudium mole. So some of the Pseudium species are in Allahabad. You know that wilt is the first incidents where it has happened or where they have find out. It is in Allahabad. So in that, starting from initial species, they were trying to uh, find out some of the solutions for this wilt. So they have all conserved all the species in Allahabad. So these are this fresh Yavitanum. So if you talk about this acute angulum, it is very resistance for waterlogging areas. So this, if you why it is having acute angulum, this will be having two here, then it will be having a opposite direction, and it will be the leaf will be very acute. If you found these leaf characters, the tip it will be very sharp. And our student Dr. Pradeep is there. From TNIU who studied here. He is one of the scientists in NPBGR, where he is a very pioneer in characterization of any species. If you want to find out which species it is there, so he will help you. In NPBGR Trishur, he is at present working. So he is the person. If you have a flower and fruit characters, so he can identify in any of the crop. He is very, very well expert in that. So this we have in IRA and IHR. Okay, these are some of the told. So these are some of the accessions where we have this. What are the genes? It is there. So in high vitamin C, it was earlier report. So now, as far as that, Alaba Safaida and Lucknow 14 is also having higher vitamin C. This big size fruit that VNR or Bihi Goa or KG Goa we called, but this one is having higher shelf life. But quality wise, we couldn't find us our traditional varieties like Alaba Safida. But we want to improve this because what is the major problem in red flesh? One of the question for one of the student. Huh? Seeds is there, more seeds is there. Shelf life, yes. Yes. When you compare to white type, it is very less, maximum 3 to 4 days we can keep. Otherwise, it will deteriorate. But normal time it can come to 5 to 7 days. But when we talk about this KG Gova or VNR, it will have in room condition it will be degree 12 days. So we want and it is like a coconut. The crispiness will be there. It is in the five-star places where we can be offered. But local we don't or we dislike this consumption when we come back to our traditional varieties. So then why we want to conserve? So normally we have more earlier we have more variability. So when suppose I like red type means I will cultivate. Then after that, if some new varieties are coming, then we will discard that. Like that, in our horticulture college, Periyakulam, near to the pond, we had a citrus collections, germ blossom earlier. Now, if you go, it is not there because when we want to have a over a period of time, when we want to get it back, and we will not able to get. And likewise in Kilikulam, Dr. Shanmusundram has worked on guava. That species I have ordered for IRI. After that, one drought came. I think Sarmasundram went to Chennai. Now all the plant have died. So he has collected more number of species, but it has not been maintained. So conservation of whenever we want that we cannot get it back, because now that policy of importing, exporting of planting material is very, very difficult of the quarantine. So whatever it is there, we should conserve. So that is one of the main aim for a breeder. As an institute, as a national depository, we should conserve all the species, but what character we want to use, it may vary. So that time we may not able to get. Now we are having as an institute or germplasm depository, we should have all this thing and we should conserve. So that's why we thought there are two methods of conservation. One is in situ field, that is field gene bank. Another one is in situ conservation, in vitro conservation. So where we can go for conservation of pollen grains, or we can some tissues we can take that and we can cryopreserve that and it. So both we are having an IAH, cryopreservation and other thing. So these are some of the main center of collections. Here I said IHR is having Bangalore, then CHSS. Most of the centers for tropical fruits, we are having both centers like Bangalore, then Lucknow. Okay, so mango, guava, and some of the field. So these are some of the characters of this species, which I am not going to read again. So when we talk about genome, or so when we want to have a one question also morning that was there, how to go for a tissue culture or a biotechnology way to develop a seedless variety. How to develop? Those who answered don't answer here. But how to go for a seedless variety if you are going through biotechnological way or tissue culture or something like that? How we can generate? 2N, 3N it is there. So you don't take the embryo. Or which part we should take then? Endosperm. Endosperm have 3N. No. So it is having 2N. So if you take that, then you will have a chances of developing a seedless. So this one aneuploid we talked about that has been developed. So normally, some cases tetraploids also there. Here, main advantage is that we have in South Indian condition we have three flowering season in like grapes. Like grapes in IHR we take 5 crop in 2 years in Goa. So it is sometimes continuously flowering now. And mango also one or two other genotypes we have identified. It is having a throughout the year we have found 1 or 2 crops. Now some of the students yesterday when we are going for lunch we had a discussion. Now for inducing of flower, so we go for culture. So culture is to be only done for off season. So when we go for regular season, then the phenology changes. And if the phenology changes, there are some problems of expression of male and female. So how to go, and it is only recommended for irrigated condition. You go for culture application and you should have a full irrigation, then only it will respond, otherwise the plant will die. So when there is under stress, normally which flower will it will produce? Male flowers. Whenever there is a stress, it will produce male flowers. So it don't have that nutrient or energy to produce a female flower. So it requires more energy or nutrients for production of the female flower. So these are some of the situation when we adapt or when we change, then that will create to the environment. So in that case, in flowering we have April 3 season. So and sometimes it is a continuous season also like grapes we can do. But for off season in mango, there are some natural genotypes are available. So my emphasis will be one of the students can have this already long back they have taken. So one of the students can take as a feature aspect for production of off-season identification, some of the survey and clone, so that we can have in Salem area it is there. Now, earlier we found that off-season flowering is specific to Kanyakumari and part of that Ramanathapuram or seashore. But now, past 1 year, I came after Bangalore. I have seen so many places. There we found out of season mango genotypes are there, not in the cultivated field. It is on the roadside and other things. So that case means it is genetically it is having a capability to identify flower in the off season. So that the collection should be done, and that has to be evaluated so that without any manipulation of chemicals, we can have some varieties which will give some yield to the farmers during the off season. So regular season, what is the problem? There will be a sometime glut or there will be a less thing. So in that time, like Totapuri or Bangalore, we talk in sometimes in Andhra. So it has come less than 10 rupees or 5 rupees. So there was a huge cry that what will farmers do? So and there is no, they are not taking even to the processing industries. So in that case, now if you go and see wherever you go to religious places, they will sell mango. From where they will get you take to Kanyakumari, you go to Rameshwaram, you go to our nearby temple. Also, you can see mango fruits. So one small cup they will give it as a 20 rupees. So if a fruit can be cut into 100 rupees minimum. So the average, if it is in availability, if you make, this is the way we have to have as a breeder or a student, where we can think of producing a variety of naturally, then it can survive into the local accommodated conditions. When we don't modify or give stress to that. So that's why I am also now involved in four crops in IHR Bangalore, but where we go for mango off season is my one of the objective. Then whatever I did in IRI, we have guava for mutation breeding. I am going for mutation breeding. Then sapota and jamun. Jamun is the same. We are also targeting for dwarfness only. That's why we asked. Okay. Then when we talk about crossing, so when we go for crossing, we need to know the timing of anthesis. This anthesis time is not very constant. It may vary depending upon the temperatures. So if temperature is medium, then it will have this 8. Otherwise, what will happen? 8 to 10. When it is a little bit cold, it will up to 10 o'clock it will take. But when temperature rises, it will still go below. Early morning, if temperatures are high, then pollen and this is it will happen very early. So it is not very constant characters, or it depends upon the temperatures. Similarly, in case of mango also, that will have pollen dehiscence takes upon the temperature. So if somebody want to do that also, it is the same, similar way. And it is, as you know, it is called often cross-pollinated. So here we need what are the problems? So here, when we talk about, we want a large area, so maintain a large area. Uh, some of the problems are there. So somewhere if we talk about that classifications and the thing, so always there is a problem. So what happens when we go for this cross-pollinated species? So it will have a not uniform. So that's why in horticulture, when we go for first cross, then when we identify, we fix a character, we fix a fruit, except banana. So we fix a fruit, fix a character, then we go for clonally multiplication of that. So that's why we don't want to go for F2, F3 generation, except papaya. In papaya only for some of the standard protocol we follow like annual crops. Otherwise, here horticulture, we find a trait, we find a character. Based upon the character, we fix it and F1 itself. Not we are not going to F2 back cross and other possible for diseases. We are not able to go to the back cross and other because it itself is a heterozygous nature. So whenever the normal that dominance characters and recessive character does not exist in fruit crops, most of the heterozygous fruit crops. So here, these are some of the popular cultivars. I am not going to read. And here some of the improved varieties where AHR has developed. This also I will just skip off. These are some of the Arka Kiran. Then Arka Purna, this is the latest variety. But here we found that it is good in shape and but we have bold seed. So normally what for a fresh consumption, we don't want to have a bold seed or not a too soft seed. It should be chewable in nature. And if you see this thing, we have a more number of seeds. So this should be less. We want, but whenever we talk about seedless, Seedless doesn't make any proper shape in guava. Some indigenous hormones developed by the seeds that is required for a proper shape, either in round or globose or whatever the original shape of the fruit. That needs some of the seeds. That's why we always say that we need some seeds, less number of seeds, and it should have proper maintenance of shape. Otherwise, if you go for a seedless, it will be misshapen. That is one of the drawbacks. Or it will be small in shape. So for that, we need to apply some of the hormones that we don't know. So these are some of the varieties developed from Lucknow. So this one is the latest variety. Lalima, I think the Maharashtra student, what is your name? Huh? Deepti. What is Lalima? Lal means what? Huh? So only because of see here, what is the main problem means in Tamil Nadu, because I am worked in North India for past 25 years, that I think that we are literally of remembering some problems. So because they give some names of their local understanding. Lal means red. So Lalima, for the student who is studying in Hindi, they will easily remember. But for us, we have to memorize Lalima, Lalima like that. We should memorize. That is the one of the drawback. And Hathi Jewel in Amla. What is Hathi? Wait. What is Hathi? Not Vangi. So there is, so because when I went to after joining this job, so I found that is Aarti. Second one, that is we are taking Aarti. That is not Hindi. Hathi in Hindi, I am asking. So now you can tell elephant. So it will come like this. So jewel, it will come like a drooping. So that variety is called Hathi jewel. So there are some characters like that. So these are some of the area where we doesn't know. We cannot recall, but that students, those who know Hindi, it will be very. So this is language also make an impact, and that's why we want uniform. We should learn some other languages for our development. Let them fight for not Hindi speaking or thing, but whenever we want to go out of, because when we are not having job, when we, I am not pro for Tamil or pro for, we want our job. So we should learn Hindi, everything we should is for our understanding. Not if I learn Hindi, I will not forget Tamil. Still we talk in Tamil only. Yes, Sanmasundaram. So whenever we see that is our mother tongue, that's why we say whenever we, the better understanding will be having in mother tongue. So that's why their mother tongue is Hindi, so they can recall. So I am not against of any language, but the how the concept we develop that matters. So we should learn whatever it is there, but for our developments. Okay. So this is Lalima. So it is red. So similarly, we have one in a mango also one variety is there. Pusa Lalima. That is similar to this color also. For some of the competitive exam, it may be so that's why I am worked on both the crop. So I am interrelating that. So this one is we have to see. Now what we have targeted is to develop a variety. So you can see this thickness. Na? See both the varieties. Where this is a Posus region. The rootstock variety we have developed. So I am a partner of this variety. I am two varieties we are there. This is LA developed in 2000. Developed in 2022-23. Now recently released only for Delhi. It will come to creep in next year. So here we found that the pulp content will be more, but the size is only around 200 gram to less than small, medium size fruit, not very big size fruit. Because when the seed is less, the fruit size is also will be medium. It will not have a bigger size. When seed is more, then it have a more bigger size. So these are some of the Allahabad Safeda. So Allahabad Safeda around 350 grams, Lucknow 49 350 grams. So here Allahabad Surka is there. Surka is of two type. One is outer side peel color is red, another is pulp color is red. Okay, so there are two types of Surka is there. These are oldest varieties. And Isar Surka. Isar Surka is one of the best variety. And here sir, Safeda. So one of the parent we have used for that is Sir Safeda. For it is already having a parent of seedlessness. Trait might have influenced in our cases. We have one of the parent for our variety as Sir Safeda also. These are some of the Rajasthan, some of the varieties there. So here, beside this area, we have some of the research are going in northeastern regions also. They have also developed some of the varieties. Meghalaya. Okay. So here, uh, which one? Ida. But it is showing some seeds are there. There are some seeds are there. So, uh, so another area is that where major concentration is there in Punjab. Okay. This color development, Dr. Muthuel, if you get this plant, you can plant it either in Kodaikanal or Ooty. So because I correlate this with the cold temperatures. Whenever cold temperatures are there, this color may develop. So that can be because Punjab is also having lower temperature during winter, Delhi, then UP. So then that it expresses major only in winter Goa, not in rainy season Goa. So it is a temperature related one. So you can, if you want to have this, we can have planted in our research station so that we can see and whether it can come better. So this is, we have also used this Punjab pink. Color is good, but at the same time we have a little bit bold seeds are there. So these are latest variety they have developed. This one. Safeda means Safed. Safed means white. Again, in that's Safed means white. Safeda. So there are some problems in crop breeding. So as we talk about, so there are some why we can go for breeding in fruit crops means we can fix some of the characters. So fixing is easy. Then especially for guava, propagation is easy. But some of the root stock we are having some problem. But other than that, some characters are there. It is easy, but we cannot study the inherited characters. But still, we are doing in IRA. One of the co-worker along with me, Dr. Madhubala, along with some of the students. So we have clear characteristics. Some of the characters I can show, like red flash will be dominant, but always not dominant. It will not express because it is dominant means it should come in every type, but it will not express. But bold seeds are dominant, so these are both are linked characters. So when we talk about these are some of the so we want for processing. Main is color for processing. So these are some of the color we extracted here when we talk about that purple or guava. So that is the juice of this one. So this is Punjab pink juice. So these are some of the varieties. So when we talk about breeding, so for what purpose we are going to take? So these are some of the characters where you can for traditional breeding. These are the characters where we can take for rootstock breeding. But I told that we are not much worried about dwarfness. We should worry about wilt and nematodes. So these are our prime area because dwarf we can manipulate with the existing varieties. So here some of the resistance to biotic and abiotic. Like major is that in biotic, fruit fly and mealybug are one of the major problems. So if you want to develop some of the things, we have some of the management practices, but still if you have able to some resistance, then it is good. So when we talk about what are the breeding methods, so introduction, then selection. So some it may repeat. So wherever I have told in elaborate, I will finish it. So survey. So wherever we start for collecting some of the species, one of the curriculum students also doing in that some estimations in Cape Gooseberry. Huh? So in that, so for that we have to collect. So we have to make a survey to every place. So when I started in 2010, so we went all over India, wherever we have a team. So some four staffs were there, four scientists were there. So I mainly concentrated on southern parts, then some eastern parts like that. But Rajasthan we had a less thing, but we have a more. So main area is for to find out already there will be some of the types that are suitable for our thing which is not available to the public. So in that case, whenever we have a like this one, Manila tamarind, so it is a selection. So Virudhana, so we should identify. So there are now some naturally existing things are there. So we have to go for that survey. Then we'll go for a collection. Then you evaluate at a particular condition. So it may be performing better than the available varieties. So in that case, you can release this better as a check. Then you can go for that. Then promising ones are selected. So another one is that once you collected this one, so there may be some deficiency or there may be some good character in one or the other variety. So what character you want to take? In that case, you have to hybrid. Like that, I took Isar Safeda. It is little bit soft also, and it was a cross of seedless. So one of the parent in our variety it is Isar Safeda. So after that, we want to have a color. So we made one uh, color flower to cross so that we can make into different colors. So like this. So here hybridization one is interspecies, intraspecies within species or variety. Another one is interspecies. Interspecies for transferring some of the resistant characters. So whatever characters I have discussed earlier. So another one is major one is that, but this is a mutation breeding. Mutation breeding is a you know? So out of 1,000 lakhs of seeds you go and treat and variations we can get very minimum. But for vegetative method, colchicine you apply, it will multiply, it will change the shapes, but it will not exist in the. We don't want to multiply. Ploidy breeding doesn't help in case of this one. And if you go for ploidy breeding, then pollen viability, stigma viability, that problem will be there. So if you have a variety which is of good characters, it will be best, but it will be very rare, and sometimes it may also revert after some period of that. It is a disadvantage of that this character may also revert. So that is the thing. But we are trying. So out of something, whether we can do or not. Then molecular breeding tools. So we have some of the tools. So where we can screen that you design a primer or something like that. So we can screen a larger population. We can reduce the shorten the period. That is called as we discussed speed breeding or fast track breeding or something like that. It can aid in that. So if you want to lack of population to flower, plant, flower and absorb, it will take years to get. So for what character you have to design a primer, you develop a molecular tool. After that, you go for a large scale screening like papaya, sex expressions. So female, male, and other thing. So somebody was discussing that in Coimbatore area, they plant 5 and 1, and they don't want to cut after growing because they are very good people, Coimbatore people. But in Tirunelveli area, we will cut people also, so we don't bother that also. So like that. So here, good people are there, so they even don't want to discard after growth. So that is a good. This belt, when we talk about this, there is a good approach. They don't want even to harm the plant where you have grown. That is a very good characters because you have grown up to 6 months. After that, you have to pull and it will dry. So why has to done that? So that is a good approach. I feel after listening to some of the some staff today, so she told that farmers are feeling like this. So that is a very correct. No, so why I want to grow up to that height and want to. So in that case, if you recommend a technology where 1 or 2 plant can be grown, so what happens if you grow 6 plant, then it will not bear better. If it is male coming, female coming, that then ultimately the whole 6 months growth will be affected. So for that we should develop some of the markers. So that only molecular breeding will help. So these are only one introduction is that it is demand, it is susceptible to Tyler and rock. So selection already we have talked. So this one is some of the area where I visited. These are the area where we see. This one we have color. And in Delhi we have one pulp, one fruit in winter only. So outside will be there and that side inside color. So here the seed will be more. So we here you can see the seed will be less. So based upon this collection, so we have made some of the hybrid. These are some of the selections already there. So when we talk about hybridization, so these are the basic steps you should know, or you will be knowing also. I will just go for. So we have to take that pollen when it is going to open. So this is the critical stage where we want to see that it is stigma is receptive now, but there is no external source of pollen contamination is there. So if you want to take a pollen parent, you cover at this stage. Before that, you don't want to cover. You, and especially for guava breeding, it is very, very easy. Numerous pollens. But when we take in case of mango, we have five, and very, very small. So crossing each one, it is very tedious. Why? What technique has come in mango? They have developed earlier caging technique. So self-incompatibility varieties with the this one we have developed. So for that only caging technique they have developed earlier. So it is very tedious. So for here it is very, very easy. You cover that and you can do. In some cases it is in some cases three flowers also will come. But in that case, what you should do only allow the center one, which is bold, and remove the side two flowers, which will be very small in size. So this will have a robust growth. So then after that, we are going for emasculation. Then once emasculation, this is one part. Next part is collection of anthers. So male covering the same, cover that male parent whatever you want. Then collect this early morning. You can collect. You previous day you can cover it, and if you don't cover also, we can wait up to this. This is, will days in 1 to 2 hours. So after emasculation, put a cover over that emasculated flower. Then you can make the pollen arrangement. So normally, if you want to store, then you can go for this method. Otherwise, you can directly collect the flower, go for immediate crossing means then, because in sometimes we don't match synchronizing of the varieties whatever we want to cross. Some will come early, some will come late. So in that case, you have to collect and preserve. So whenever it is ready, then you can make a cross. So for this purpose only, this another collection is there. Otherwise, if you are instantly having both the parents, then immediately you can take a fresh flowers, then you can follow it and cross. And after this, you have to back for 3 days, minimum for 3 days, because the stigma receptivity is up to 72 hours. So after that, you have to, and this butter paper are there, it should have a ventilation. So otherwise, it will have rotting or moist will be there. So depending upon that, or in some area where temperatures are more, it will drop due to heat stress. So these are the things you have to do. So these are some of the examples or whatever I did. This is for how to go for. It is only a numeric value where I want to stress upon is that how we go for after making crosses. So what are the parent you have taken? In that case, different parents you have taken. So make a label. Once this much hard work we are doing, so labeling and documentation is more important, because otherwise we will miss the track. Okay. So in that case, what are the crosses we made? Make a list of crosses. So in that we have to make what day which flower you have crossed. Then after some days we have to take for the uh, setting, fruit retention. So how many days? So normally when temperature rises or there are some natural stresses, it will be there. So after a fruit, you have to go for one irrigation for both guava and mango. So there should be minimum stress. Otherwise, that abscisic layer will be there and there will be a drop. So if you want, and only single flower and terminal that bold size flowers are allowed, and very minimum flowers in mango for a bunch, we go only 5 flowers, and that too in terminal it will be good. So normally if you see the bearing habit in mango, so only the terminal flower panicle it will be. So we select in the terminal only, and 5 to 6 flowers per bunch in case of mango. But here we can allow one or two or per branches. So once it is matured, so we can give protections. Okay. So this is my I am myself in 2012, I suppose. So we have developed this block. So we need a big trees, it will not be useful. So same thing, we have to go for a minimum space so that we can cross hybrids and cover that, and the operation will be easy. So these are one of the type, not the same crossed one. So after that, we collect the seeds. So there is another thing that seed germination is also a problem. So for that, what you have to standardize with a normal seed treatments when to grow. And normally fresh seeds are more having germination than stored seeds. So after immediately after taking this, you have to go for some of the hypochloric acid, 2%. You have to give that treatment. Then that case germination in guava it will be better. So that it took 3 or 4 years for me to standardize this germination time. And probably this 2 seasons are there. So winter season we don't grow it immediately. We will wait for some more temperatures to rise. Otherwise, you have to grow in a poly house. So that's why in North Indian cuticle project. So there we need that temperature will be low temperature. So that time it will take. So you have to grow in the poly house or protected cottage. So then we go for a placing of the seed. Then once it is grown, if you are not condition is not favorable, then you can go for a poly tube, poly bag, or if it is you are ready to field preparations are ready, then you can directly take it from pro tray. 2 months minimum, 2 months record, not less than that. 2 months growth we have to put it. So these are some of. Uh, I have some more photos, but I am not having at present in IAHR. So I came and transferred some of the data. I left it in Delhi, so I'm not able to show you. So these are some of the hybrid lines, 8 to 10 years of crossing. So we planted, then it is this tree is around 8 to 10 years. So we maintain a separate block of hybrids. Normally now guava breeding is a little bit easy because once after sowing the seeds, it will come to flowering up to 18 months. Earlier, 3.5 years it took. Now I have observed some of the lines where it come to flowering in 18 months. So in 3 years of time, you can characterize one tree. So that is one of the advantages now in Go. So these are some of the process. This is one Punjab pink and Alaba Safeda. So Punjab pink is red flush. So but it has come red. But in that case of here, this black or purple guava allowing that while hybridization slide, I have one another white next to white one flower is there, no? That flower. But we got the work is done by Dr. Madhubala, my colleague, who is studying patterns in guava. So normally, what is this chart? What is this color chart? Huh? Where it is? Who is the authority of that? Normally, we call it as a royal color chart. Where authority? Royal means London. Okay, they have developed this one. Now that time it was 30,000, and now this color chart is around 60,000 rupees. So only they are providing. Otherwise, initially when I did in Bikaner, that time you don't have this color chart and other thing. But I asked to do for scoring of color. What I did, I went to paint shop. They are having different colors for selling that paints. So I took it, I compared, and I told this is the color. So some of the things when we have to do something, we have to have some ideas and other things. So, but in IRA we have, and IHR also we have. Here also we might having in floriculture division normally we are having floriculture we had earlier because they are more having colors for flowers. But good. So these are some of the things. Now there are some fruit fly less of varieties. So this we have to use for this breeding problem. Another problem in wherever tea estates are there in southern parts, we have this tea mosquito bug is one of now latest problems. So I already told this red color is dominant over there, and it is governed by monogenically. Red flush is heterozygous, and another one is bold. It is dominant over this one. Flesh color and seed size are linked. It is linked. Okay. So these are some of the things. Some of the Parthena carpi we can exploit in that. So these are some of the things. Replied you can use for that. So another work is that we want to suppose after collection everything, we go for a molecular characterization. So these are some of the primers we used. RAPD also we used, but as we discussed, RAPD, if I do once, it will not be repeated again. So we don't go for RAPD for any breeding work. So we want SSR marker. So we developed we identified some of the markers and we have screened some of the gem blossom. So after screening, no, so this is another one important marker which has a good result, but it is very, very costly. So for a student research and other thing, we couldn't not. So see, we can do a very hi-fi research, but we want money. Money is ultimate for doing any research. So once we do this one, and we invest around 2 to 2.5 lakh per student for molecular work. But at the same time, when we don't publish that, we are not going to. But for publication in a good journal, we want to again invest in publication charges. So for a better work for a student, minimum 5 lakhs. Next, I will show one of my PhD student. We have published in some good journal rating. Why I am saying means those who do a better work, our aim should have a better journal. Here we have what is the NAS rating for PhD? 6, more than 6. But why to stand on 6? Let me publish on 10 and more than 10. If it is better for submission, one paper can be 6. Rest of the paper you can improve and you have time. So it will better for your students' career. At the same time, we have some credit to the university also. So in IHR, we are ranked number 1 in all ICR institute, not even horticulture, in a number of ICR institute, because most of the journals publications we have different ranges, more than 15. Some scientists will be having some projects they are having. So more than 10 we are having. So all over the now they are going to divide by the staff. Earlier staff is more. So we got now it will be accountability for per scientist. Means 200 papers are there, 200 scientists are there, then it will divided by 200. Then it will get 1. Suppose what happened? Solapur is there, pomegranate, NRC grapes are there. So they are having 15 to 20 scientists. So they cannot compete with us. We are having more than 200, 300 scientists. So they want to rationalize. Now this year onward they was revised. But even then we will become first because our contribution is more. So because when we want to work in molecular or tissue culture, that needs more investment. That's all. So these are some of the things we did. And after that, we go for dendrogram based grouping. So it has 3 major clusters and which are the varieties are related. So this much jam blossom earlier we had in Delhi, but here we have more than this in IHR band. So among the species also we want to find out which is having close correlation with some of the genotypes. So these are some of the things. So this work we did for quality of parameters and we want to do for vitamin C because vitamin C is some varieties more, some varieties less. So we can estimate by biochemical estimation. Then you might have asked why you want to go for molecular marker. But this molecular marker, where it can be useful? Gene editing. Suppose you want to have this one for gene editing process. So this we can find what are the pathway, which gene is located on which chromosome. Then you can go directly transfer from that gene desired variety. So these are one of the objective. We took it in 2023. My student completed a PhD. We took it in 2020, I suppose. But this for this we have in Dr. Muthu. Here there is no funding for NAHEP, National Higher Education Program. No funding from ICR. No, irka illa. So there in ICR system in IRA we have National Higher Education Program. So in that my student he got applied and we have 2.5 lakhs for this work, which was very helpful in part time. So he did some of the first we go for characterization of this. Already this is the topic of my students. So we were doing so along with the student. I am also standing. This is 2020 to 21. So we are estimating all the collected genotypes. We had around 29 or 30 genotypes we had. So first we go for a biochemical one. We want to screen because we have that much. For each primer, it cost 15,000 rupees to sequence. So we don't want to go at a initial stage. We don't want to screen that much. It will go into lakhs of rupees. So for that purpose, initially we went for a biochemical screening. Then we found out the lowest and highest. Then among that major highest one, and we want to have a two different gene to find out which one is having less gene for ascorbic acid, which one is higher gene. So in that concept, we did some of the work. So this was Allahabad Safeda. It is having more vitamin C. This Panch Prabhat, even one red type is also. So here lycopene is more, but we have this is white type, but it is having less vitamin C, around 100 grams something. This one has 160 to 180 or 190 grams. So as you know that we have four pathways for ascorbic acid. So these are some of the four pathways. So these are some of the enzymes where these genes are involved in ascorbic acid biosynthesis. I'm not going to read. This you can take it from your head if you want. Huh? So some of the questions, there are some in silico. What is that in silico method? Already there will be there. So in that we have to identify some, because in guava there was no earlier work on these characters. So already some similar vitamin C is estimated in some other crops. So we have to you have to go use the software, blast it, and find a sequence. Then you have to match like that. So this is the work, very tedious work. 3 years, if you are very sincere enough, it will be done. Otherwise, it will not be done. So my student is very, very sincere, and he did. So these are some of the transcript data available. Then you have to go and do it. Then ultimately, we found a complete genome is for that crop. So after that, these are some of the method. So in how which method we are going to, and we specify the season. Why? While estimation, we specify the season because we have two seasons, rainy season and winter season. Winter season is little bit drought, means we have less water. But in rainy season, we have more of water content. So that quality may vary. So whatever character we do, or we should mention the season, temperature. Suppose you are saying room temperature for storage studies we are going. What is the room temperature? That is more important. Room temperature today it may be different. In April it may be different. In November it may be different. So whenever you write any paper or any article or any work, so specify the particular temperature or summer season so that we can have correlate with the studies. So that for that betterment. So this is the sequencing method. We have used this one. So these are some of the molecular procedure. I think most of the thing you know. I want to explain each and everything. So these are some of the process where we did. We took some constant. After that screening, we have go for contrasting genotypes. We go for a transcriptome. Then we took some transcript. Then we designed some of the primers. So after synthesis of this primer, then we go for a standardization on this. Then we go for amplifications. Then sequencing. So some of the sequences we have taken. Then again we will go again to whole genome data. Then if it is not completing again, then again we come for a primer design. So these are, if you are very lucky enough, it may get it very in short time. But it needs sometimes electricity failure. Sometimes you may run the gel, all of a sudden electricity will be going off. So again the process has to be done. So it is very, very tedious process. Molecular work requires some patience, but it will have we can have a good publication. Without molecular work nowadays, there is no paper accepted in a better journal. So for that only we tried with that student, and it is a good result we got. So these are some of the things. So here you can see some of the marker we identify with the DNA of some of the varieties where we are going to use. So after this, my student work, there are some more student work. I thought since I am going to share with you this PowerPoint, I have not put much of the result because we want to publish in another one more journal. So for that purpose, I have not put more. And we have designed 16 primers, and we have deposited in a national for plant biotechnology. So this is my student contribution in that. So we also have first time in Goa. So this is some of the achievements. So some topics we do like that artificial intelligence some student is doing. So that is having now present in horticulture or anywhere, artificial intelligence for next 5 or 10 years, that will have a good scope. So whoever do, it should be because now last before 10 years, we don't know. But it has happened. Nobody thought of having a mobile time. So in our studies, we don't have much facilities, but we had a good teachers that time. Still, you are having a better teachers here. And TNAU, we should have, I think, what is your name? I forgot your name. She has a different college exposure. So she may be having. So that's why in earlier, our batch, Dr. Shanma Sundaram is here. We are the last batch to have the ICL fellowship in the same university. So after that, they told no inbreeding depressions are there. To get fellowship, we should to move to other institute. So we were the last batch lucky enough to join here. Then we went out and other things. So most of the students went to GKVK and some of them went to Hyderabad and other things. So if you have go to different institute and in national wise institute, GB Bandh University is one of the best university. PAU, then TNAU. These three are sometimes will be ranking will be first, second, third, and these are the top universities. And some of the colleges in Maharashtra also. That in Eclipse Center is Raguri is one of the center where they have developed a variety called Bagua in pomegranate, which is still a ruling variety. But Solapur Lal they have replaced now, which is with biofortified varieties, which is picking up well. So there are some significant contribution like so these are some of the QTL and mapping. What are the things has to be done for related to Go? I'm not going to read. So in other cases, mutation breeding we can go for in vivo and in vitro, and some of the physical mutagens like gamma chamber, X-rays and other thing. But normally we go for common chamber. In chemical mutation, you can go for EMS. For cultivation only, you can when you want to make into double haploid, you are taking breeding, then you make it to diploid. For that purpose, it can. Or some of the, if it flowers, then it will be good. So these are, so not much work I have done in mutation. I have given some treatment. We have observed some of the mutants. So we only fixed not to F2, F3, only find it in F1. And similar way, we are doing for Jamun in same here in Perioglum, they are doing in Jamun for darkness. IHR also we have for Sapota and Jamun we are also doing. Both mutation we have done in zero point 0.5 EMS for Sapota. Then you have for Jamun also we have both physical and chemical mutation we have. So these are one of the latest where we want to have a dwarf trees. That's one of them. So another thing is that biotechnological techniques where you can go for somoclonal variation. So it is good for a breeder and not good for the multiplication. If you go for multiplication, you want uniformity, but variation can be useful for a breeder. So and haploid breeding is the latest thing where we can go for a homozygous planting. Not much I have covered after that, whatever. So these are some of the area where you can go for. So now major problem is that one is processing. Another one is quality, lycopene and other thing you can go for. Next one is the major area we can go for shelf life. So how to increase the shelf life. So that is the next. So that we have already found in some of the Thai guava type. So that has to be find out the gene or used for crossing material, then that can be done. So another one for wilt resistant roots. So already I think some of the progress we have done in IHR Bangalore. So that, but the main problem is that for multiplication. So if you standardize that, it will be a better one. So that is the thing. And I think TNA is also, our division is also doing good work. Dr. Sanmu Sundaram, we had some visit. So I hope you should first publish that, make a record that you have done first. Because otherwise they will come and they will take your idea. So that being a colleague also as a friend, but you should not wait for somebody. First, whatever you discover, make a record, publish in a popular article that we have done the first. So that is the thing wherever sometimes go for in food crops, patenting is very, very difficult. But if you want also do for some of the technology, that has more value. Okay. So otherwise, these are some of my visions, and I want to acknowledge all these contributors. So these are molecular biology scientists in ICR NBCR, sorry, NIPV, and our earlier director and present director, and my student, especially two students, one is Shaili Kumari, and the one is Nayanti. Because of them, I had a good research paper also. And funding by ICR for my student. So any interaction, any question, anything you have idea, what I know I can share. I cannot tell I know everything because it is a learning process. I can learn some of the students qualifying by also something new I think learn. So somebody wherever we go, we have to travel. So if you know something, I will learn from you. If I know something, I will tell to you. So this is my point.